Now we are going to learn a few tips and tricks to improve your experience with XRNA. First, we have three hotkey combinations which act as shortcuts for UI interactions. To use these hotkey combinations, you must first click somewhere within the XRNA program. If you attempt to use these hotkeys before clicking into XRNA, your browser will attempt to interpret these commands in ways which have nothing to do with XRNA. Number 1. Ctrl-O launches a menu for opening files inside XRNA. Number 2. Ctrl-S launches a menu for saving files externally. Number 3. Ctrl-0 resets the viewport. None of these hotkey combinations require you to have a certain tab open. You can use them at any time to accelerate your use of XRNA. Let's zoom in. Another trick which may improve your experience is the right-click and drag function. When you are using the Edit tab, the Format tab, or the Annotate tab, you can skip manual selection of a constraint. Instead, simply right-click and drag across as few nucleotides as possible to select your desired substructure. In response, XRNA attempts to infer your desired constraint. Then it populates a left-hand menu. XRNA accomplishes this by examining the nucleotides which you clicked and dragged over. Then it iterates from the most specific constraint to the most broad, attempting to match all dragged-over nucleotides to a single constraint. Therefore, the more nucleotides you select, the broader a constraint XRNA auto-detects. Now we will demo a couple of examples. Dragging over a non-base paired nucleotide results in the single nucleotide constraint. Dragging over two mutually base paired nucleotides results in the single base pair constraint. Dragging over a contiguous run of base pairs results in the single helix constraint. Dragging over a large selection of unrelated base pairs results in the RNA molecule constraint. Thank you for watching. See our other videos to learn more.